Hello everyone and welcome to the latest Genome Giants interview. Today we're going to be joined by Canadian scientist Stephen Scherer. So before we get stuck in and look at your career, Stephen, if you could just introduce yourself and tell everyone a little bit about what you do as well.
Yes, I'm Steve Scherer and I'm the Chief of Research at the Hospital for Sick Children and a Professor of Medicine at the University of Toronto. And I've directed the Human Genome Center here at SickKids, called the Center for Applied Genomics, for the better part of 20 years now. I'm formerly a PhD trained genome scientist, and probably the first of the generation of scientists that would kind of dub themselves genome scientists.
So if we just take it back to the beginning, you were born in Windsor in Canada. What was some of your fondest memories growing up, and what were you kind of like as a child as well?
Well, for those of you who don't know, Windsor is right across the border from Detroit, Michigan, the United States. It's the automotive capital of Canada, just like Detroit is of the world, arguably. So it's a very blue-collar town, a very humble upbringing. I had three brothers. My parents were working class parents. We had a completely carefree childhood. I was the second born son, spent much of my time trying to play catch up with my brother and overtake him. And I often joke in my presentations to my students about the contributions of genetics and nature and nurture, so genetics and the environment, that the most important event in my life was probably being born second, actually, in a family of four boys. So my environment was to work hard, to push and try to catch up with my older brother, and also beat up my two younger brothers too. But no, we had an incredible, we lived at the edge of a park and we spent all of our time outside exploring, playing sports. It was just a great childhood, and that was really the major influence in my life.
And when did you first kind of strike up an interest in science?
Yeah, this is really interesting because, as I said, most people in my high school would not have even thought about going to university. Their parents worked in the factories and things. And certainly, I thought when I was going to high school, I knew you could be a doctor, you could be a lawyer, or you could kind of have some type of a trade or something like that. But I didn't even know that this thing called science existed. So for me, it was in grade 11, reading. We got to select one book in grade 11 biology, and the book that I found in the library, probably because it was the one where there were 10 copies, was James Watson, The Double Helix, which is the description from his viewpoint of the discovery of the structure of DNA. And that really captured my attention, and it was the major reason. My grade 11 biology teacher also was outstanding. And I think he caught that I had a special talent in looking at things from a different point of view, and encouraged me. And then I took science courses in grade 12 and 13 and right into university. But it was that reading of that book, that first exposure to some biology. It was the first time really I thought about it in a molecular way.
Yeah. You completed your science degree at the University of Waterloo. What was your time like here, and what made you want to select kind of biology and chemistry?
Yeah, so for those of you who don't know, University of Waterloo would be kind of the equivalent of MIT in the United States. It's a very technology heavy, it's an outstanding teaching school, engineering, math, science, and they have a cooperative education program there. So you typically go to school for 4 months and then work in your discipline for 4 months. So your undergraduate is 5 years because you're spending a lot of time out in the workforce. And for me, I had to do it because I wanted to go away to university and I had to pay for myself. So I had to work. But at the same time, I also, having a very practical background, I wanted to have a job when I graduated. I really didn't know anything about this endeavor called science. I didn't know when I went to school, you could do a thing called a PhD. I really didn't know what that was until maybe second year university. So it was for practical reasons. And, we went around, looked at several different schools, and you can apply in our province in Canada for 3 at a time. So I applied to the 3 that I wanted to go to, and Waterloo was the one, and I got in this co-op program. So this is the reason I went there. And then I have to say, I was very, very fortunate. I had, and I'm happy to talk a lot about this, but I had, I think it's 6 work terms, all in different areas that were just outstanding. The first was at Agriculture Canada. It was outside in the county or outside of Windsor, Ontario. I got really lucky and had my first 2 jobs there when I worked on virus genetics, virology of plants. So I was working with virus DNA and RNA back when I was 17, 18 years old. This is really unbelievable. This will be a theme that comes up in my answer to your questions, is to try to expose young students to things as early as possible, challenges. And then I got a job actually with the Royal Canadian Mounted Police. That's the equivalent of the FBI in Canada. And I was in Halifax on the East Coast, and I was involved in setting up the first statistics for DNA testing in Canada. This is in 1985. So again, exposed to just an incredible environment. And then to top it all off, my last few work terms were in Ottawa at the National Research Council, which is really the top place for certainly the physical sciences. And in Canada, after the war, when science was set up, funding was set up, it was first founded out of the National Research Council at the Sussex Drive laboratories, which are still there in Ottawa. For people in Canada, they'll know what this is. This is a big thing. And I got to work with a group there, which was a leading edge molecular biology group. Kind of grew out of the era of DNA synthesis. Michael Smith, Canadian Nobel Prize winner, he wasn't there, but many of the influences came. And earlier, there's a reason I'm going to tell you this, is in physical chemistry, there was many advances there, including Gerhard Hertzberg, who eventually won the Nobel Prize, and many of the subsequent Nobel Prize winners in physics came out of that laboratory. And the director was E.W.R. Stacy, and I didn't get to meet him, but I learned a lot about him at the time I was there. And later on in my career, I was so honored I won the Stacy Prize. So anyways, a lot of it goes back to the history of the people I met there. I was a summer student in 3rd year, and I remember there was a group of us who worked very hard, but we played really hard too. And Professor Hertzberg, the Nobel Prize winner at the time, came up when we were laying on the lawn outside on the Ottawa River, and he came up to 2 of us, and we had had a rough night the night before, and he said, you guys need to be working harder if you're going to be successful in your science. In other words, get back to work. So it was, meeting these giants of science early on in my career really had a positive influence, and understanding the history was very important.
Yeah, I feel like you had such kind of a broad experience, and the exposure that you had. How do you feel like that then went on to kind of shape your career?
Yeah, it's exactly it. I was, always looking for a new challenge and very curious. And I was offered to stay in Ottawa to do my PhD, and it wasn't going to be enough for me. I really wanted to work in medical research, and the work they were doing in Ottawa was more physical, as I said. So I went to the University of Toronto and interviewed, and there's a whole story behind this. We often joke I didn't get in till really late in the summer before I started in September. I think I got in and accepted in June or so, because they lost my application 3 times. Maybe they were intentionally losing, it just wasn't that strong. I don't know, but I since that time, a few years back, I become a distinguished university professor at the University of Toronto. This is the top ranking. They only give a few out to the professors. And in my acceptance speech, I made reference to that it was great to win this distinguished honor, but you lost my application 3 times. But anyways, so when I came here, they have, again, a great system where you have to rotate through different laboratories before there's a mutual agreement of the one you're going to finish, do your dissertation, thesis, research in. And I had heard about this place called the Hospital for Sick Children. Of course, they had these ads on TV, and it was really seen with the Stacy Institute and at the NRC. It's really one of the top places in Canada. And I said, there's no way they're going to take me. And I went to a lecture there. This is really quite relevant to our discussion today. In September, and Ron Wharton, who was the head of genetics at SickKids, he'll be known to many of you as one of the individuals who discovered the Duchenne muscular dystrophy gene in 1985, 1986, was also found by Kunkel's laboratory in Boston. And Ron had just come back from, really, it was the first Human Genome Project organizational meeting. I think it was in France, the first significant one. You'll see the pictures of the old group of scientists, and Watson was there, and many of the leaders. And Ron was one of the Canadian representatives. So, and he gave an overview, and just sitting there and knowing about the double helix and things. And I said, this is what I want to do. And then on the way out of that lecture, Lapchi Choi, who turned out to be my PhD supervisor, and of course, in 1989, led the discovery of the cystic fibrosis gene. And I should say this was 1987. He walked out and he knew who I was. I mean, I couldn't believe it. And he kind of cornered me in the hallway. And he said, why don't you come and do a rotation in my laboratory? Okay. And I said, well, yeah, for sure. This would be great. And I said, later, many years after, and we've become friends, after I got my PhD, and I said, you remember that day you cornered me? How did you know who I was? And he said, well, it was the first year I actually looked through all the student applications. I needed someone who had a background in yeast genetics. And I had done that in Ottawa and NRC. I was doing gene cloning in yeast. And because there was this new technology in 1987 that came out from David Burke and Maynard Olson in Washington, where cloning large DNA molecules in yeast. And it was really the first primary technology used for mapping chromosomes. And at that time, the cystic fibrosis gene wasn't identified yet. And Lapchi, including in collaboration with Francis Collins at Michigan, were developing methods to move faster along chromosomes. So Lapchi said, you got to come and do this yeast cloning in my lab. So again, being exposed to this, and one of my heroes and mentors came and talked to me. It was great. So I ended up at SickKids, and I've been here ever since. And I've always looked to try to do my research where I can have the most impact. And this is the place for me that I could always have the most impact. And that's why I've stayed for the better part of 30 years now.
Yeah. During your time as a research associate, you're actually involved in studies of mapping human chromosome 7. What was this experience like, and how does that kind of compare to our capabilities today? Yeah,
it's really important for the students to go back and remember the history of the Human Genome Project. And again, I teach this in my courses, although the amount of time I spend on this history gets less and less. But a message certainly for the younger people is learn the history of science. There's a lot of great lessons there, and I'll try to bring a few of them up. Yeah. So for me, so there were stages of the Genome Project. One was the gene mapping stage, and there used to be these human gene mapping workshops, and there was a chair of each chromosome, and these people would get together with their big binders. There was no internet or anything, literally in suitcases full of papers, and meet at different places around the world. I remember in London, it was 1990 or 1991. I can't remember the year. And that was the first one I got to go to. And they would collate, there was literally like the X chromosome or chromosome 7, maybe had 100 genes on them. They actually put pictures up and pin them. It's kind of like pin the gene on the donkey, on the chromosome. But anyways, this was the serious state-of-the-art genomics at the time. And then that evolved into the technologies for large DNA cloning and pulse field gel electrophoresis analysis and things, and mapping, or RFLP genetic mapping. So each chromosome had teams working on them. And in Toronto, because the cystic fibrosis gene was on chromosome 7, and TAC-MAC's work on T-cell receptor was on chromosome 7. There was work on P-glycoprotein on chromosome 7. We kind of claimed chromosome 7 as Canada's chromosome, and we started doing the mapping, building out from the cystic fibrosis locus, to be honest. So Lapchi had, of course, found CF, and I was a student, and he was famous traveling around. So I kind of just took over the project as a graduate student. But it was great. I got to go to all these meetings. I was at the very first Human Genome Project meeting in Cold Spring Harbor. And again, it's very important. Right through the 90s, it was all mapping based. And then Jonathan and Eric Lander's group at MIT started really in France and in the United States to lead whole genome type approaches around genetic markers and large scale fingerprinting and things. And these kind of maps provided the basis to hang the scaffolds of sequence that started to come from Celera's whole genome shotgun approach, and even the Genome Project's clone-based approach. They still needed to have these genetic markers. So through the 90s, chromosome 7, we had literally hundreds of collaborations going. And you can see on my CV the number of genes. We had identified more than 20 different disease genes using all of these mapping reagents. So if someone mapped a gene on chromosome 7, they would typically come and talk to us in Toronto. We would try to help out. Yeah. So again, just to come back, it was the exposure. I got to meet these incredible scientists. They're really the best scientists, I would say, of that decade were going into genomics, because it was big science and it was something new, and people were curious. And there were some naysayers, of course, but it was just such an exciting place that I think the best talent, they wanted to be at the cutting edge, and that's where it was.
Yeah. You also made significant contributions to the initial description of copy number variants. Would you be able to kind of expand on this time and what actual the response was from the scientific community at that time as well?
Yeah, so I just started my own laboratory in the late 90s, and we were just kind of finishing the chromosome 7 project. And our approach for the chromosome 7 was to actually use all available information. As a scientist, you have to do that. You should use all data, positive and negative data, to try to come to the best conclusions. So we were using data coming from the public project, data coming from our own sequencing, all the mapping, and we also had access to the Celera sequence. And we were collaborating with Craig Venter's group and combining the data. So we published it's not a highly cited paper, but I think it's a very seminal paper in 2003 that was the first integrated chromosome 7 sequence of the genome. And in fact, it's essentially the approach that people are using now for whole genomes. But anyways, there were a lot of lessons learned that came out of the paper, but along the way, we did some very early sequence comparisons, assembly comparisons of the Celera data, the public data, and there was a lot of overlap in our own data. And we found that there were a lot of inconsistencies. Okay, and while much of the world was writing off these inconsistencies as mistakes in sequencing or assembly errors, we could start to see, because we were using another technology called chromosome microarrays, that in fact, these were likely to be structural changes in chromosomes that missed the resolution of karyotyping, so it was smaller variants, and they were larger than you would see typically using Sanger-based dideoxy sequencing. And I'll come back to this in a second. And so that was in the early 2000s. But really the reason was we were the first lab in Canada to have access to whole genome microarrays. So these are the 1 million base pair resolution back microarrays. So there was one clone roughly every million base pairs spotted on a glass cover slip. Okay, so there was a company called Spectral Genomics in the United States that made the first microarray slides like this, and the scientific aspects were run by a Canadian who I bumped into at a meeting, and another scientist who was also a Canadian scientist, Charles Lee, who was in Boston, was using the same arrays. And so Mansour Mohammed, who was the Canadian in the United States, connected us all to workshop, and we kind of combined our data, and we could show that we were seeing the same thing because we were using the same technology, that there were these unexplainable differences in copy number in a lot of different regions along the chromosome. So when Charles and I saw our data, we had each done about 25 genomes, and these were expensive experiments to do for a young laboratory. I'm happy to talk at length about that, but the reason I could do them is I had a philanthropic donation from a very wise donor who was supporting our autism research. So cut a long story short, in the early 2000s, we saw these, and because the Genome Project really had missed these dimensional changes, it was really missed because of the result of how the sequence came together. And it was one of these rare events in the history of science, again, where the discovery happened at an in-between phase. So in most discoveries, if you're looking at things of size, there's a continuum. You tend to find something that's small and then extend out to something that's big, or something that's big and small. If you think about physics and the universe, for example. And here, because the in-between technology, the microarray technology, didn't exist, karyotyping 60 years ago saw that 0.6% of the population carried chromosome changes, the general population. Sequencing, and largely by the Genome Project, but even earlier, looking at the gene level, found that there was about one nucleotide change in 1,000 or so that was polymorphic. But there was no technology to look in between. And really, it was that, people forgot about the history. Well, there's no technology to find this. Of course, you're not going to discover this variation. So because we were the first to do these experiments, certainly in Canada, there were others in the States, and we're very open-minded to this. We saw the data first, and we made we discovered it, and we combined our data and published it. And Mike Wigler's group in Cold Spring Harbor was using a different microarray technology that he had co-developed, and they saw it around the same time using different samples. So these papers were published in 2004 in Nature Genetics and Science, respectively. It was good they were co-published. It helped people believe it, but our paper took well over from start to finish, I can't remember now, but well over 12 months to get through peer review. And the peer reviewers just didn't believe it. And I remember spending hours and hours and hours arguing with the editors. I remember one August afternoon at our cottage back, this was Zoom arguing with Nature Genetics and doing multiple experiments. And Charles' lab in Boston was doing the same and getting more data. And then making presentations, even to the people here in Toronto. I knew how important this was. We developed a database for genome variants. And I said, we have to start looking in mouse. We have to start looking in rat. It's going to be there, right? And nobody believed it. The clinical lab didn't believe it. So my PhD supervisor, Lapchi Choi, had just left at that time to go to Hong Kong to be the president of the University of Hong Kong. He believed it. In fact, he pointed out to me that I told you that already 5-6 many years ago. You just didn't listen to me. And he's actually right. But we had the technology, and it was the open-minded aspect of science. And just to reinforce, now we know when you do so-called end-to-end chromosome sequencing or whole genome sequencing, copy number variation in the context of structural variation, it's the major form. You see roughly 30 million base pairs of heterogeneity compared to the reference per genome, compared to the 3.2 million SNPs or so. So this is a major player in genetic variation, and it's like anything early on, it's hard for people to believe. So
yeah. You've since gone on to discover loads of disease-associated copy number variants. What is the kind of significance of these findings, particularly your work related to autism spectrum disorder?
Right. So it was really in parallel. I'd mentioned how expensive it was to do these chromosome microarray experiments in the early 2000s. I remember the first batch of arrays we bought. It was upwards of $2,500 an experiment, and it wasn't unusual for us to be running $50,000 experiments in a week, and most of them failed, really. So for a young scientist to hedge this, it was in a new lab. We were very well funded on Canadian standards, but like nobody could have afforded this. So we were actually running mainly samples from individuals with autism because they had funding also to do that. And if you look in the 2004 Nature Genetics paper, in fact, we explained that we actually, some of the samples were from people from autism. And what we found was that there were a higher rate of C and Vs, both de novo, so spontaneously occurring, and rare inherited in people with autism compared to general population controls. And this was, we were doing our own study. We were studying about 400 families from Canada. We published that in 2008, but we were also part of the International Autism Genome Project that was using more of a linkage and genetic marker approach through the late 1990s and early 2000s. But in fact, they were based on Affymetrix microarrays, and we could call CNVs from those too. So in 2007, as the consortium, with our group leading this, a lot of the CNV work, we found a lot of de novo. We studied 1,600 families. This was unbelievable. This across the world, and found roughly, I think it was 7% of the families compared to the 1% in controls that had these de novo changes, and they often hit the same genes, and these genes were involved in synapse development and communication. So, so that was kind of the first really entry point, looking at, en masse globally rare variants in a particular disorder. And I've collected now as a consortium here in Canada and across North America, well over 4,000 families, and we've replicated this using higher resolution technologies. In fact, it's interesting that CNV is playing a major role in many neurodevelopmental gene disorders, modeled after this early work in autism. Many other studies in schizophrenia and others have found similar things. And the reason is that 70% of our genes are expressed in brain development, and brain genes tend to be much longer with lots of different isoforms. And I think the reason CNVs have such a role is you need to knock out a lot of the different isoforms. Otherwise, if you have a point mutation, you can kind of skip over that exon and get to still some form of functional protein. But the CNV is bigger, and it may knock out the entire gene, the whole entire locus, or multiple exons. I think that's probably the reason.
How is the work kind of from the Autism Genome Project being translated into the clinic, and how is that informing management of this condition as well?
Yeah, so we moved from microarrays. We've been very well funded and supported by Autism Speaks, which is an international organization. But we've been supported by the United States and Canada and philanthropic donors and granting agencies to successfully use the different higher resolution technologies, from microarrays, higher resolution microarrays. We pretty much skipped over exome sequencing. We moved into whole genome sequencing, just knowing we knew we had to have all classes of variants, in particular the structural variants for autism. So we started that in 2012 already. It was pre all these other projects you heard about. We were paying like $7,500 a genome. I spent most of my time fundraising. But anyways, so now we've collected over 4,000 families across Canada. There's more across North America, but I say the Canadian because we've enrolled these families. All of the clinicians know the families. We communicate with them in regular workshops, and we ask them the question you just asked me, such as, why should we do this research? And the answer from the families was, we want to know why. Why did autism come about in my child or my brother or sister? We want to know what we can do to help. This was the second answer. How can we help in the research? And third was, and this has been replicated in different interviews and different workshops we've had, what can we do? What do we do now with this information? So to cut a long story short, for roughly 20% of the families, we can start to answer those questions. If we do a whole genome sequence, there's roughly 100 different genes or CNVs that are identified through worldwide efforts that are clinically signed out as being involved in autism or the related medical complications. And in many cases, the information used is a confirmatory diagnosis to explain that it's genetics. It had nothing to do with the environment or the vaccines that you took, or what you did or didn't do during your pregnancy. So, and often that can also inform on genetic counseling for family planning or older siblings, for example, on likelihoods. We're starting to see now for some of these very highly penetrant genes, things like synaptic scaffolding protein 3, SCN2A, a sodium channel gene, there's a genotype-phenotype correlation, which may inform on things like anti-epileptics. Epilepsy is a big player in autism. 50% of autistic individuals have seizures and anxieties and things. So we're starting to see that it's influencing medications that exist. But for autism, there's actually no medications. There's therapies, there's behavioral therapies, but there's no medicines to treat the core features of autism. So most importantly, I think, is we've identified pathways now. All right, so we don't have answers. We have answers for maybe 20%. Other 80% is much more complex, very genetically heterogeneous. But we've got good pathways now. Lots of great animal models, cell-based models, and many biotech and pharma are interested in this again. And there are actually some new drugs going through clinical trials on the patients. We've done the genetics, and this is relatively new. So I was very fortunate to be in the lab, as I said earlier, in 1989, where the cystic fibrosis gene was identified. And I don't think any of us could have really predicted there would be, everyone wrote it in the grants in the first period, you get the gene, you'll get diagnostics, of course, that's immediate. But then there'll be medicines. And now for cystic fibrosis, there are effective medicines for specific mutations. So predicting the same will come for autism, absolutely it will come. And it'll come faster. I don't know when, but it'll come probably in the next decade.
Yeah, that's exciting.
Yeah, it's very exciting. It's incredible. It's really incredible.
Yeah, I think it would be good to kind of discuss some of the contributions that you have kind of made to genomics within Canada. And one of those is founding the Centre for Applied Genomics at the hospital for sick children. What was this kind of journey like, and what are some of the significant contributions that the centre has made to the field of genomics?
Well, forming it was a necessity. We were always developing the technologies. I talked about large cloning, and then eventually moved into sequencing. It was more of implementing. Early on it was developing, co-developing it, taking other ideas, making them better. And then when it came to sequencing, I mean, these were things developed in industry that we implemented, but then made them better for our own circumstances, more efficient. And as I said early on in this interview, the most important advice I can give to young trainees is you need to be close to the technology so you can see the data for the first time, right? Or you need to have the technology. And now it's, in a genome center, you need to be in the genome center so you can see the data coming off the machines for the first time. It's relatively easy to make a discovery if you're the first to see the data and, you have some good ideas. So that was really the reason we did it. And in the Canadian scheme, for us, many of these technologies are very high throughput, and we didn't have the funding to run them at full capacity in our own lab. So we opened it up. And the model was we ran a service laboratory where my lab was the biggest user. And this has been consistent. We support well over 800 different laboratories. In some cases, there could be 10 different people in the lab using it. So, billing and keeping this technology. And that's how the Canadian funding system has also developed around putting infrastructure in place. There's the Canada Foundation for Innovation, and then Genome Canada. So it was out of necessity to get the data and also keep the machines running and be able to pay for the whole thing. And it worked. And then what we did just more recently, I think it's about 5 years ago, is we linked the three major sequencing centers in Canada. So at McGill Genome Center, led by Mark Lathrop, the Vancouver Center, led by Stephen Jones and Marco Mara at the BC Cancer Agency, and then our center here at SickKids. We're linked together through this Canada Foundation for Innovation Major Science Innovation Grant called CGEN. And now we have this big infrastructure grant, and we work together and we share samples and data and things. It's really quite spectacular. So again, it was, you have to figure out how to make it work in your own environment. I think that's going to be a theme of genomics globally, going worldwide. The power of genomics is global. It's the study of the human population or any other population, animal species. And the key, I actually think the key discoveries are going to come in studying these unique populations now. So other countries, other groups that are starting out, you have a chance. You have to do it your way. That's going to work for you. And again, look back at the history of other projects. I think there's a lot of lessons to be learned.
Yeah. Alongside your colleagues, you also launched the Personal Genome Project Canada in 2012. What was this like, launching this project like, and what's the significance of this project as well?
Yeah, so a little bit of the history again. My kids get to hear this at home and they just walk out of the room. So, but the story was we were working with Craig Venter's group after he left Celera and started his own institute. They were doing his genome. It was, of course, a lot of the data was the Celera data, but they enhanced it and published his own genome in 2007. And it's probably still the best reference sequence, to be honest, but it was based on Sanger sequencing. And because of our work in CNV and the previous chromosome 7 collaboration, he had asked our group to do this CNV annotation and kind of medical interpretation with their group. So we got to see, firsthand what the impact of having a full, quote unquote, diploid genome sequence was. So this was through 2000 up to 2007. And then George Church had started this project in the United States called the Personal Genome Project, where the idea was, the genome is a composite of our humanity. It's not really it is your genome, but it's so shared. Why don't we just put them all out there as long as you consent to do that? And George and his wife came to Toronto and his daughter and gave us talk. And I remember talking to them, said, well, we should do the same in Canada. And so they provided their consents. We Canadianized them and developed a project that kind of went in parallel. And it was really, really important. And the motivation for the project, and I was funding this off philanthropic donations we had, was to really learn all of the lessons that we needed for things like the Autism Genome Project and cancer projects later on the Canadian standard, because consenting here is different than in the United States. Healthcare is administered provincially, not nationally. There's all kinds of nuances, and every country is going to have. So we wanted to learn all these things, and we also didn't have a Genetic Non-Discrimination Act at the time. So we had to learn how to develop the consent to patients. We had to figure out the technologies at the time that was still $10,000 a genome at this point. Using Celexa data wasn't that good, to be honest, et cetera, et cetera. We wanted to check all the boxes and learn each step along the way. That was the project. And we did that. So in the end, we were very ambitious goals. A national paper in Canada, Globe and Mail, ran ads. We had thousands of people register. We enrolled through a private healthcare clinic here called MedCan, which were incredible collaborators. We did the sequencing, fully consented. It is the most open release, I think, anywhere. You can go to a website now and click on the data and download it. You don't have to go through any data access committee or anything. It's all been consented this way. But it was tough to do a lot. We didn't do as many samples as I would have liked. But the most important thing is we primed the system here, and I think shared our data and our experiences. And when Canada kind of matured to the point where it needed a Genetic Non-Discrimination Act, we were able to provide the data that, look, we've got this out there and it's not that harmful. In fact, no one has had any problems so far. And because we had these experiences when the legislatures and politicians and government agencies and who are putting this bill together, we could provide the scientific support for it and brought the Canadian community on side. And it was really that event that pushed this bill across the threshold. So we now have a very comprehensive bill, and in a large part, I would say, it was built on the experiences of the Personal Genome Project. So it's one of those things that I'm really proud we did it. At the time, it was scientific, but I could see there would be an endpoint. And now looking back, it was really the fact that it helped get this bill passed into legislature in Canada. So we continue the project, but we go deep. We go small and deep. We're really doing a few samples and people, a lot of genetic count. I should say that we published our first paper in, I think it was 2018, in the Canadian Medical Association Journal. And we published it in the Canadian Journal because we wanted to have impact in the Canadian system. And it was amazing because they used to still print the journal back then. They don't do it anymore. But that article is on the cover. It landed in, I think it was 40,000 doctors' offices the next day. And, in the old days, you would get all your information sitting in, both the patient and the doctor sitting in the doctor's office. And they learned about genomics through this paper. It was on the cover of all the newspapers. And I don't think anything had more of an impact, maybe COVID now, educating Canadians on the impact of genomics than that paper, even though it's only been cited a few dozen times. Mm-hmm. So, yeah. So anyways, that was the rationale. And so we continue that. But all of our consents later coming from the Autism Project and many of the other Canadian projects in part were built on that initial project, which built on George Church's project. So it was a great example of collaboration.
Yeah. I mean, if you could take a look at your career, were there any major missteps that you felt that you learned from and helped you throughout the rest of your career?
No, I think we wrote a paper, and again, it just recently came out in Human Molecular Genetics. It was it's the 30th anniversary of that journal, and I was thinking about I'm going to write something a little bit unique, and it was essentially the lecture I give the first year genetics students is you want to, when you're doing any experiment, you don't want to rely on one data source, right? And especially if you're going genome wide. And I learned early in my experience, if you just use one technology, look at anything, you're probably going to get the wrong answer or not a complete answer. So I always lecture, you need to have at least two technologies, and it just plays larger into life is you need to always have a plan. You need to have a plan or backup plan or a way in, a way out. And so that's my major advice. And I was able to learn that early on. And it was really through success, but more so failure, actually. If you do 1,000 experiments and you're really at the cutting edge, probably about 995 of them should actually fail. And that's how I train my students. And genomics has kind of normalized everything. A lot of the experiments work because it's high throughput technology now. It wasn't always that way. So anyway, so it's good to fail as long as you can extract something interesting out of the data. And that's really been the foundation for most of our discoveries, right? And looking at outlier data. So no, there wouldn't be anything I would do different. I've been very fortunate. I think challenge yourselves and don't just take the normal route. It's such an this is the most exciting time in biology, even more so than the early stages of my career. You can read, write, and edit genomes, and essentially you're only limited by your own creativity and the ethics boards that you need to work through to make sure the experiments are socially acceptable. But it's been a great career so far, and I hope I have another kind of leg in it. But.
Outside of your career, what do you like to do in your spare time?
So again, that's evolved over over my different decades of my life. I was very athletic as my earlier years, playing highly competitive to the national level, hockey and baseball. And then when I went to graduate school, I threw my competitiveness into that and biking and things. And now I have two high school age children, so I guess that would be my hobby. And we have a cottage in Northern Ontario, so kayaking and fishing and like every good geneticist, usually after they retire, but I'm an avid gardener. many of my peers, they say, what? And it's really, it's the one thing I can go out and do and relax. And that's when I get all my new ideas. And I say, if you do a good job, your plants actually listen to you, and most people at work don't listen to you. So.
That's funny. If you could turn your career into a book or a film, what would you name the title? Oh,
geez. I think probably something like, Bruce Springsteen's Never Surrender. You really can't give up. If you have an idea, you'll hear this over and over and over again from scientists. If you have an idea, it's usually right. You just have to figure out how to make it right or how to get to that point. And that's the characteristic of being a scientist, in particular a genome scientist, I would say. In this era is you just got to kind of stick to your ideas and go, and eventually it'll work. And sometimes it may take decades, but it'll work. So that's why I would pick Never Surrender.
No, I like that. Thank you so much for talking to me today, Stephen. The work that you do to propel kind of genomics within Canada is so amazing, and I'm excited to see how it evolves as well soon. So
Thank you
so
very much.
Hello everyone, if you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website, frontlinegenomics.com. I hope you enjoy.